Amino acids must be modified inside a cell before they can be used as a carbon and energy source. Some bacteria can remove the sulfur atoms from sulfur-containing amino acids, producing hydrogen sulfide, using enzymes called desulfurases. One of two media can be used to determine whether a bacterium can produce hydrogen sulfide. These media were developed to differentiate members of the Enterobacteriaceae. Both media contain peptone and ferric ion. We'll inoculate peptone iron agar first. Label one tube for each bacterium to be tested. Flame the needle until it is red hot and let it cool. Pass the mouth of the tube of inoculum through the flame and then use the sterile needle to obtain some inoculum. Remember to pass the tube through the flame again before recapping. Then, stab your needle into the agar. The needle should touch the butt of the tube before removing the needle from the tube. Inoculate the other tube with the other organism. Flame your needle and let it cool. Repeat the inoculating process using the second organism. Obtain some inoculum and stab your needle into the agar. Flame your loop after inoculating. Incubate the tubes at 35 to 37 degrees Celsius for at least 48 hours and up to 7 days. Any sulfide produced by the bacteria will combine with the ferric ion in the medium, producing a black precipitate. If an organism can reduce sulfur to hydrogen sulfide, the hydrogen sulfide will combine with the iron to form ferric sulfide, which is a black precipitate. Any blackening of the medium indicates the reduction of sulfur and is a positive result. The uninoculated control shows the original appearance of the medium. The precipitate forms in the butt because hydrogen sulfide formed at the surface diffuses to the air before reacting with the ferrous ions. The other medium, triple sugar iron agar, provides a little more information about a bacterium, sugar fermentation, and hydrogen sulfide. TSI slants are inoculated by stabbing into the agar to the bottom of the tube and then streaking the slant surface. We're inoculating different bacteria into each tube. Flame your needle and let it cool. Use your needle to obtain the inoculum. To inoculate the slant, hold the tube at an angle, stab the needle into the agar until the needle touches the butt of the tube. Then pull the needle out of the agar and streak the surface of the slant. Repeat this procedure using a new inoculum and slant each time. In your lab, you will be instructed on the number of tubes to inoculate. Remember to flame your needle and let it cool after each inoculation. The tubes are incubated for 18 to 24 hours at 35 to 37 degrees Celsius. Longer incubation of the TSI may result in bacteria using up all the sugars and producing ammonia from peptone. This will change the pH indicator back to red, masking any fermentation. Observe the TSI slant for growth and a black precipitate indicating hydrogen sulfide was produced. TSI contains phenol red indicator and 3 sugars, glucose, lactose, and sucrose. A bacterium that ferments only glucose will show acid in the butt of the tube. The bacteria quickly use the small amount of glucose and start oxidizing amino acids in the peptone for energy. This produces ammonia, which increases the pH, and the indicator in the slanted portion of the agar turns back to red. Carbon dioxide, or hydrogen gas, may be produced in the TSI agar matrix, forming noticeable cracks or fissures in the tube. This may be in addition to hydrogen sulfide gas production. This bacterium produced excess gas, causing the agar to lift from the bottom of the tube. If a bacterium ferments lactose or sucrose, more acid is produced, and the slant also turns yellow. The bacterium in this tube ferments glucose and at least one of the other sugars. This bacterium ferments glucose, at least one other sugar, and produces hydrogen sulfide. Remove the labels and dispose of the tubes properly.